Good afternoon. I'm Matt Roth with Baylor College of Medicine, and I welcome you to today's presentation as part of the Extracellular RNA Communication Consortium webinar series. The ERCC is an NIH Common Fund program that works to advance the science and research of extracellular RNA. Stage 1 of the consortium started in 2013, with the second stage starting with the kickoff meeting in September of this year. The consortium hosts monthly presentations on a variety of research topics, and I'm very pleased to introduce today's speaker, Dr. Siyang Zheng, Associate Professor of Biomedical Engineering and Electrical and Computer Engineering at Carnegie Mellon University. The title of Siyang's presentation is Extracellular Vesicles for Cancer Diagnosis and Therapeutics. Siyang?
Thanks for the introduction and also the invitation. I'm glad to share some of our recent results on using extracellular vesicles, or EVs, for cancer diagnosis and therapeutics. First is my disclosure slide. So I'm currently Associate Professor at Carnegie Mellon University. I'm also a co-founder of Tactics Diagnostics. So here's the outline of my presentation today. First, I will give a very brief introduction about the EV-related work in my research group. And then I'll talk about two stories we recently finished. One is develop a lipid biopsy as a non-invasive cancer diagnosis based on EV isolation. And then I'll talk about using EV membrane camouflage nanoparticles for intracellular and systemic protein delivery as a cancer therapeutic approach. And then finally is my conclusion. Okay, so first, just as a brief introduction, I will show some of the works we did in this EV-related domain. On the cancer diagnosis side, we invented a technology we call lipid nanoprobe as the rapid way to isolate total extracellular vesicles from samples, such as blood plasma. And then more recently, we adapted this technology into a microfluidic device. Separately, we also have run some clinical study. Basically, we compare cfDNA with DNA derived from extracellular vesicles. We found the amount of DNA is lower in the EV fraction, but actually the quality of the sample in terms of the CT value of the PCR is actually slightly better for EV DNA. And then on the therapeutic side, in one study, we actually use lipid nanoprobes to put aptamers on the surface of the EV, and also we developed a method to generate artificial EV using cell membrane. And then combined with these two advancements, we actually developed EV-based nanoparticles loaded with chemotherapeutic drugs for cancer treatment. In another study, we actually loaded EV membrane with new nanoparticles incorporated with proteins.
Getting up and.
So today I will focus on the lipid nanoprobe story and also on the nanoparticle approach for protein delivery. So first is a project we finished a couple of years ago on developing a lipid nanoprobe for EV isolation and cancer diagnosis. So currently, cancer diagnosis at the molecular level is mainly accomplished by tissue biopsy. However, we know there are a lot of issues with tissue biopsy. First of all, it is an invasive process. And then because of that, the accessibility of the tumor can be an issue depending on the disease. Also, the cost and the turnaround time can be a problem. The other major issue I see is the spatial variation and the temporal variation of the tissue biopsy. It's just because we cannot do tissue biopsy for every part of the tumor, and also during the therapeutic process, it's very difficult to do tissue biopsy many times. And then sometimes the amount of tissue is a problem for tissue biopsy, especially if we want to send out the tissue for next generation sequencing. Sometimes that can be an issue. So we see there's a new opportunity for new technologies to replace or at least complement tissue biopsy. So the particular technology we have been working on is a group of technology collectively called liquid biopsy. So as the primary tumor started to metastasis, there are a lot of tumor-derived material that will turn out in the blood circulation. One type of this material is circulating tumor cells. Another one is a circulating tumor DNA, and then the third one is more recently extracellular vesicles or exosomes. So in my research group, we have been working with circulating tumor cells and extracellular vesicles. Okay, so this slide probably is familiar with most of the audience in this community. So it's just the introduction of extracellular vesicles. These are sub-micrometer vesicles. They're released by almost all the cells we know. They play an important role in intercellular communication. Structurally, they are lipid bilayer enveloped vesicles containing rich contents such as proteins, nucleic acids, and metabolites. And then these vesicles can be heterogeneous in size, composition, and how they are generated. And as we know, there are a lot of different names for those kind of vesicles, but nowadays we collectively call them extracellular vesicles. Okay. So here's the overview slide of the lipid nanoprobe technology we developed. So this is a nanomaterial-based EV isolation technology. There are two components in the system, at least for the initial technology. One is we call the labeling probe. So you can see it's a lipid molecule. So we can use a different type of lipid, monoacyl, diacyl, or for example, cholesterol-derived molecules. And then there is a PG linker. So we can change the length of this PG. And then at the end, there's a biotin tag. And then the second molecule is a capture probe. Basically, it's an avidine surface conjugated magnetic nanobead. So the way this nanomaterial system works is we first mix the labeling probe with the sample. What happens is this probe will spontaneously insert it into the membrane of the extracellular vesicles. So basically, we tag those vesicles in this step. And then we use the magnetic bead to pull those vesicles down. So here is some electron microscopy images of the capture probe, the vesicles before they are captured and after they are captured. As an engineer, we actually optimize this process. For example, we optimize the amount of labeling probe, the incubation time of the labeling probe, the incubation time of the capture probe. And also what we found is when we switch from cell culture derived EV to plasma samples, we actually need to increase the labeling probe by almost order of magnitude to account for the lipid binding proteins within the plasma sample. So in the end, what we have demonstrated is we can have about 84% capture efficiency from cell culture suspension and about 50% capture efficiency from the plasma. One good significant advantage of this approach from our point is this is a very fast process. We can run this process within 15 minutes without any bulk equipment. So because of this, it's ideal for relatively small sample volume, from microliter up to a couple of mil. So we think it's good for basically clinical plasma samples. And then what we do is we actually use a variety of downstream analysis methods. So for example, we can extract the DNA and RNA and run electrophoresis. We can use RNA-specific fluorescent dye to stain the RNA inside those vesicles and quantify the RNA amount. We can also run protein immunoassay and basically showing the protein content of the vesicles is consistent with the cells where they're divided. And then, of course, we can run a Western blot. We also use TM and particle tracking analysis, nanosite, to characterize the size of the particle isolated from using this technology. We also use a variety of different EM technologies to visualize those isolated vesicles directly. In particular, we also run some proteomics study to try to compare our method with ultracentrifugation and also a reference paper published by Harris Group on PNAS in 2016. So what we see is the top 100 proteins from Vesiclepedia, we can find about 89 of those proteins using our lipid nanoprobe isolation approach, compared with about 76 using ultracentrifugation and 96 in the reference paper. We also did a little bit further study showing that the proteins from the proteomics study have are highly associated with exosomes and lysosome fraction. If we basically just characterize these proteins according to the cellular locations. So when we test the DNA content, we found it very interesting at that time. So we, again, in this case, we studied the copy number variation of the DNA content after the lipid nanoprobe isolation. We compare that with ultracentrifugation and also the genomic cellular DNA. So what you can see from this result is that basically we don't see any difference between those three approaches from these three materials. So basically what that means is if we can really assay for the DNA content of the vesicles, it actually can represent the DNA content of the cell where these vesicles are divided. So encouraged by this kind of a study, we actually run a pilot clinical study. In this case, it's about 20 plasma samples from non-small cell lung cancer patients. And then what you see here is we are actually trying to detect EGFR mutations and KRAS mutations, which are the two therapeutic relevant mutations for this disease. And then what we do is we actually try to detect those mutations from the EV sample. And for most of the cases, we also have the result from the tissue sample. There are some cases that the tissue sample among this is not enough for NGS sequencing, so it's not available. So at the beginning, we actually just use a single sequence to see if we can detect anything. So we actually, I think we're pretty lucky to detect just one mutation from these samples. You can see this is a KRAS G13D mutation. The majority of the allele is actually wild type, but you can see there is a peak for this mutation. So we're very encouraged, but then our clinical collaborator told us there's probably more mutations. So what we did is we used more sensitive detection technology in terms of the real-time PCR and also mutant-specific restriction, enzyme digestion, and massive PCR. So in the end, we actually detected more mutations from this pilot sample pool. However, there are 3 cases in which there are mutations from the tissue sample, but we were not able to detect from the EV samples. So at this point, we realized the mutant allele fraction of these within these samples derived from plasma can be relatively low. So we actually need to use more sensitive detection mechanism. So one outcome of this study as a follow-up study, we actually tried to make this principle into a microfluidic device and also use more sensitive digital droplet PCR for the detection. As you can see for this diagram, what we do is instead of using 2-component system, we basically conjugate the lipid nanoprobe on the surface of a microfluidic device. To improve the capture efficiency for this particular device, we actually generated nanostructured substrate and then conjugate those lipid nanoprobes. And again, we use off-chip digital droplet PCR as a more sensitive detection mechanism. So to create nanoscale the surface morphology, we actually use technology we developed. So basically, we have a thin metal film on silica wafer, and then we do a thermal annealing with the presence of hydrogen. So the thin film metal will form nanoparticles on the substrate. And then we basically use this, we can characterize the size and the interparticle distance of this substrate. And then what we do is we use this substrate as an etching mask to etch the silica surface. So we use RE dry etching, and then we can control the etching condition to achieve certain desirable surface morphology. So this is the fabrication process. We deposit iron, and then we do thermal annealing. We run RE etching, and then we dice the wafer. We remove the etching mask, and then we started to do surface functionalization. First, step is the finalization, and then we functionalize the surface with the lipid nanoprobe. So basically, in this case, we don't have the second molecule, which is the capture probe. We directly functionalize the lipid nanoprobe on the surface. And then finally, we passivate the surface to minimize non-specific binding. So after fabrication the substrate, we also bonded this device with the PDMS micro mixer chamber. So this is well known in the microfluidics field. We are using a hydrogen structure to mix the sample inside this device so that the sample will be able to in contact with the surface. So here is the top view of the device. So we actually did a little bit further study to optimize the lipid nanoprobe. So what you can see here is we use a different lipid head in case of monoacyl, diacyl, and the cholesterol. So we can see cholesterol actually in this particular case has a better binding capability. And then we also changed the length of the PEG linker. So the shorter the linker, the better the labeling efficiency. So after we did the optimization, we can show the vesicles can be captured on the device. We actually optimize the flow rate versus capture efficiency. This is a continuous flow device. we have shown that actually the optimal capture efficiency is around 10 microliter per minute. So at the lower flow rate, what happens is this mixing is not as efficient, so the vesicle does not really reach the surface. At a higher flow rate, the shear stress becomes higher, and also the interaction time between the vesicle and the surface is shortened. So that's the reason we have an optimized flow rate. And to compare with the nanostructured surface with just a planar surface, the nanostructured surface does about 3 times higher capture efficiency. And then for the detection, as we indicated, we need to improve the mutant allele fraction. So we adopted digital droplet ddPCR technology. So what you see here is the axis is the spiking percentage, and then the y-axis is the detection. So basically what we show is for the mutant allele fraction, which is the mutant copy number versus wild type copy number, we can achieve about 0.005% if we spike in genomic DNA directly into plasma. If we spike EV into plasma, it's about the same order of magnitude. We can achieve 0.1% mutant allele fraction. So here in this study, we did a little bit like a pilot study. In this case, it's the top 4 are samples from patients, and then the bottom of 2 are from healthy donors. So as you can see, these are 4 patients with pancreatic cancers, and then these blue dots are the droplets with the mutant copy, and then in each individual of the green dots are wild copy. The orange copy we think are actually contains both the mutant copy and the wild type copy. So what we see here is we can see a wide range of mutant allele fraction within these samples. In this particular 4 cases, it's from about 22% down to 0.4%. So that's again highlighted the importance of detection sensitivity after the EV enrichment. So I like to switch gears, talk about the second story. This is using EV-related materials for drug delivery for cancer therapy. So the purpose of this technology is to allow us able to deliver proteins in a systemic way and then into the cells. So this is a very challenging task. The reason is there are a lot of engineering challenges and biological barriers. So first of all, the drug we want to be able to deliver are proteins. So the proteins needs to maintain their 3-dimensional structure. They're relatively sensitive to the environment. So the proteins can be degraded, denatured during the nanoparticle construction and also in vivo transportation. And then it's actually challenging to load the protein in a very efficient way into the nanoparticle construct. But this can be important because in general, we want to have a higher loading efficiency so that we can have a lot of those protein drugs, at the same time minimize the material of the nanoparticles to minimize any potential systemic toxicity. The other thing is, as proteins, if it's introduced from like foreign source, it's possible these can evoke immune response and a clearance. The other thing is we want to do this delivery in a systemic way. So ideally, we want improve the delivery specificity and minimize systemic toxicity. And then the last one is we want to deliver the protein into the cells. So as we know, cellular uptake of proteins is generally very poor. So that's one of the major issues. Okay. So here's the slide of the overall approach. So what we do here is we actually use a relatively new nanoparticle construct, in this case, metal organic framework. So you can see this nanoparticle synthesis is a self-assembly between a metal ion, in this case zinc, and a small organic ligand. And then we actually found it's possible to actually include proteins into this self-assembly process. So the protein is not really on the surface of the particle, but really inside the particle. So because of doing that, we can significantly increase the loading capacity, and then we can minimize the use of this ligand and metal ion so that we can reduce the systemic toxicity. The proteins is actually inside this nanoparticle is actually protected by the nanoparticles. So the proteins are not accessible to this protein drug. And then we can achieve intracellular release because we designed this nanoparticle in a pH responsive way. Basically, if the pH is lowered, this nanoparticle will start to disintegrate so that the proteins will be released. And then to improve the systemic circulation time and also minimize phagocytosis and provide targeting, what we do is we use EV membrane to envelope these nanoparticles. In this particular case, the extracellular vesicles we use are actually the same cells derived from the same cells we generate this tumor. So we actually collect those vesicles, we remove the internal containment and only harvest the membrane, and then we use the membrane to envelope the protein. The reason behind this is we want to take advantage of the so-called homotypical effect. So basically, these vesicles are derived from the tumor, so they can home back to the tumor. So that basically means this vesicle membrane has a higher affinity to the tumor where it's derived. And then we use this kind of a particle as a therapeutic approach to treat tumors. So this slide showing some basic characterization and optimization of the MOF-protein nanoparticles. So you can see we can actually change the organic ligand concentration and also the protein concentration to control the size of the particles. In general, we are using the particle size from 50 to 100 nanometer. And then we further characterize this metal MOF-protein nanoparticles using infrared spectrum. And also we use nitrogen absorption desorption to investigate, to basically confirm this is a porous nanoparticle. We further validate its pH responsive release. So at neutral pH, within 24 hours, is less than 25% of the proteins are released. However, if we increase the pH to about pH 5, which is about the pH value of endosomes, so you can see within several hours, this protein can be released. And then we use a different TM method to visualize the nanoparticles, the EVs, the EV membrane, and also the particles enclosed with EV membrane. And then we use a confocal microscopy, basically in this case showing the co-localization study, showing where is the protein, where is the EV membrane, and then when we merge them together, we can actually show this kind of EV enclosure can achieve about 97% efficiency. And then this is slide basically showing the uniqueness of the EV membrane. In this case, the EV membrane before and after coating on the nanoparticles show difference compared with the total EV protein. And then, of course, we use nanoparticle analysis to investigate the size of the particles before and after EV enclosure. We also studied the zeta potential, basically showing after the membrane enclosure, the surface potential becomes negative, which is consistent with the negativity of the EV membrane. In this slide, we are showing these EV enclosed nanoparticles reduces surface protein absorption. In this particular case, we are using fluorescent IgG, and we are comparing with the MOF nanoparticle, the nanoparticle coated with the EV membrane, and also the nanoparticle coated with the liposome. In this case, it's very clear that after the coating with the EV membrane, the IgG absorption is significantly reduced. We further studied this using a mouse macrophage line, and basically in this case, we show that after the nanoparticle is coated with this EV membrane, the uptake by macrophage is significantly reduced compared with the particle without coating and also with liposome coating. To show some of the potential targeting effects, first we did some fluorescence study. So we actually use fluorescent labeled EV derived from MDMB231 breast cancer cells, and then use the same EV and incubate with other cell types. Some of them are other cancer cell types or neuron cells or other healthy cells. So we can show in this case both with the fluorescent image and both cytometry that the more EVs actually can be delivered to the original MDA-MB231 cells. And then we also use the same cell line, but use a different like protein, the MOF nanoparticles, MOF nanoparticles with the EV membrane, and MOF nanoparticles with protein nanoparticles with the liposome membrane, and showing the delivery is more efficient if we envelope the MOF protein nanoparticle with the EV membrane. We further studied the internalization of these particles, and then using different kinds of endocytosis inhibitors, and then showing basically this is the dynamin and the cholesterol-dependent endocytosis. So after all this in vitro study, we did some in vivo study in a mouse model. So in this particular case, the first study we did is we tried to study the protein accumulation through systemic delivery. So we delivered the nanoparticle construct through tailwind injection. And then we use MDMB231 cells to generate this tumor model. So there are three cases we tested. The protein here is ICG labeled gelonin. So gelonin is a very generic plant toxin. So basically, it's toxic to pretty much all the cells. So the first group is just inject this protein directly, and then the second group is use the MOF protein nanoparticles and then enveloped with the EV membrane. And then the third one is without EV membrane enclosure. So we monitored the mouse from 1 hour to up to 72 hours. As you can see, we see significant accumulation of these particles even after 72 hours for the case of nanoparticles enclosed with EV membrane. And then this is the quantification of the data. And then further, we use this construct as a cancer treatment study. So what is showing here is in the we first generate a tumor model, and then we give three doses on day 0, day 2, and day 4. And then in this particular case, the negative control is DBS. We also give the animal, in one group of animal, we give the protein directly. In the third group, we give the protein nanoparticle construct, and then the final group, we give the animal with protein nanoparticle construct with the EV membrane. As you can see, the nanoparticle enclosed with EV membrane is significantly better than the other groups. So here is a more quantification in terms of tumor weight after we sacrifice the animal. When we studied the tumor tissue using the cell death assay, we show in the last group the MOF-protein nanoparticle with EV membrane closed group, we have a significantly higher cell death in this group. That's correlated with the previous slide. And then when we see the major host organs, we compare the nanoparticle with EV group with the PBS negative control. We don't see significant difference. The same is true for the animal weight. So in this case, we see the systemic toxicity is definitely manageable. So here's my conclusion. First, EVs in blood plasma contains rich information for disease diagnosis. So hopefully we show for the lipid nanoprobe approach, we can use that to isolate total EV efficiently and then definitely from clinical plasma samples. We can detect EV DNA mutations in isolated EVs from clinical samples. And then in the second story, I showed EV camouflage MOF-protein nanoparticles can deliver therapeutic proteins into cancer cells chemically. So hopefully through my presentation, we showed micro and nanotechnologies can provide technology and method innovation that propels medical discovery and hopefully eventually leads to a revolution in medical diagnosis and treatment. So here's my acknowledgement slide. On the left hand is my group members and alumni. Gong, Yuan, Zhigang, Wenxing, Marisa, CJ, Laura, Ichiu, Wenlong, and Mackenzie. These are the people who contribute to these two stories I talked about. And also I'd like to thank my collaborators, Dr. Tangablani, Xinliu, they provide with some of the clinical samples. Mr. Alberts did some bioinformatics, and Dr. Wei Mao help us with some of the biological studies. And also I would like to appreciate funding from lots of different resources. Okay, here's the end of my talk. Thank you for your attention. If you have any other questions, please let me know. And you can also send me emails for questions or comments later. Thank you.
Siyang, that's a very nice presentation. Thank you.